Okay, part two. Actually, this is more like part 6 or something, I think, at this point, of inheritance. So let's talk about the behavior of chromosomes and Mendel's laws, going back to the earlier part of the chapter. So in 1866, Mendel published his work that he had done with pea plants. Pea plant experiments, let's say. And the world kind of went, oh, that's nice. Moving on. And so then later in the 1800s, other scientists, figured out mitosis and meiosis. Of course, at the time, they didn't realize that these two things were related to one another until a handful of years later in, say, around 1902. Someone finally realized, oh my gosh, these match. These two things. And so this led to what's known as the chromosome theory of inheritance, which has got two kind of parts to it. That genes occupy specific positions that are called loci on chromosomes. And two, that the chromosomes undergo segregation and independent assortment in meiosis. As we've already talked about. So if we look back, you'll see that Mendel came up with the law of segregation and said that gametes carry only one allele for each character. So he found this, or he came up with this concept first, and then it was confirmed later on when they figured out meiosis during anaphase 1. Here we see that the two homologous chromosomes are separating in anaphase 1. Okay. And then the other part, Mendel came up with this again earlier, that the inheritance of one trait has no effect of the inheritance of another, so that the two traits are going to separate or independently. And then if you remember this, when we talked about meiosis, we showed that the orientation of the chromosomes can be, they're independent of one another. They're not reliant upon each other. They can either line up blue, blue, red, red, or blue, red, red, blue, like that. So this is confirmed in metaphase 1 of meiosis, the law of independent assortment. Okay. And we saw that when we talked about the pea plants, where they could either be yellow and round or green and wrinkled seeds. But what they saw is there's all different combinations of yellow and wrinkled, yellow and round, green and wrinkled. All the different combinations exist, right? Because of independent assortment. So hopefully that's a review. Because now we're going to talk about how sometimes they don't sort independently. So again, there's always an exception to the rule. And we found, or people have found, that some genes are in fact linked to one another. So you're going to see more commonly those two traits together. Okay. These are some experiments that were done by two scientists, Bateson and Punnett, the guy who came up with the Punnett square. So what they did is they had a F1 generation of a flower that had, so again, these are bred from true breeding varieties with purple flowers and long pollen granules. And so we're going to call these phenotypes big P and little p, so heterozygous rather, and big L, little l. So if they mated these two purple flowered with each other, you would expect you should tell me at this point. This is a dihybrid cross. So in the F2 generation, you would expect a 9:3:3:1 ratio of the different combinations of this, right? The purple flowers with long pollen, purple with let's say short pollen, a red flower and long pollen, and then a red flower with short pollen. You would expect 9:3:3:1 ratio. What they actually got, though, was, well, here I'll actually just put down the numbers. 284 of 284 of that combination, 21 of that combination, 21 of that combination, and 55 of this combination. And this ratio ends up being more like 12:1:1:2, so like totally off base, right? Way not what should be predicted by Mendel's Law of Independent Assortment. So they realized something was up. So they had to come up with an idea, and what they found was that these two genes are actually linked to one another, and they're on the same chromosome. On the same chromosome and close together. So therefore, they tend to be inherited together. So in the case of our purple flowers, we'd say maybe the gene for flower color might be here, and the gene for the pollen size would be right next door to each other, rather than being on traditionally. So this is a linked genes. And then just to compare, let's if they're not linked, let's say they're the pollen shape or pollen size is on that chromosome, and on this chromosome they're on separate ones, so that when the gametes are made, they would sort independently like normal. Here, because they're on the same chromosome, they're going to tend to be inherited together. But they did find some combinations. They weren't all purple long and red short, right? So there were these combinations here. Between the flower color and the shape of the colony. So you did get some mixing up, right? Where did that come from? Can you think why? Where do the alleles get swapped over? Crossing over in meiosis. So let's draw. Oh, that's terrible. So if we had the flower color gene there, and let's say this was big, what do we say, P? Big P. And if it were crossing over, with this one is little p and little p. Depending on where the actual crossing over happens, little l, if they happen to cross over right here, then the p's and the l's will swap. If the crossing over happens down here, then these will be inherited together, right? So it depends on kind of how far apart they are from each other. If they happen to be further apart, and P's down here, and L is way the heck down here, then the crossing over, if it happens anywhere in here, then they're going to swap around, right? But if they're right up next to each other, the chances of them, so let's look at an extreme example. If they are literally right next to each other, the chances of the crossing over happening right there is like next to 0, right? Here, there's a much higher chance of them crossing over somewhere in here and mixing up. Here, they're kind of close together, so there's going to be a small chance, but there's a possibility, right? So this change, or the distance here, is going to change what's called the recombination frequency. Let's see. This is the percentage of recombinant offspring. And we can use this to figure out where genes are located on chromosomes by doing experiments with, say, fruit flies, which people have done. And we can form what are called linkage maps. So you use the recombination frequencies to literally map out the location of genes relative to one another. Okay. So let's take let's say fruit flies. We'll use an example. If we have a female that has a gray body and long wings, and so her genotype is big G, little g, big L, little l. And we're going to mate her with a male that has a black body and short wings, whose genotype is little g, little g, little l, little l. Now, again, this is just like the yellow, green, wrinkled, round piece. So with a dihybrid cross, normally you would expect a 9:3 to 3:1 ratio if you, well, not in this particular case, but you would expect a certain ratio based on a Punnett square, right? So what this experiment showed was that, let's just say 968 of them were the offspring. Were gray-bodied with long wings. 944 were black-bodied with short wings. Okay, so these are just like the parents. So these are the parental types. But they also got this combination of 206 gray bodies with short wings and 185 black bodies with long wings. So these are the recombinant types. Okay. So if we add these up, we have 391 recombinant types out of a total of 2,300 total. So if we do the math, 391 recombinant out of 2,300 total equals 17% are recombinant. Okay. So then we can start looking at other genes that are on the same chromosome as the wing size and body color genes. So you can look at things like eye color or the length of the antennae, I guess you would say. What else would we look at? So if you start doing these recombinations, and let's say the recombination between the body color and eye color recombination, let's say, is 9%. And the recombination between antennae length and eye color, or I'm sorry, body color, is also, say, I don't know, 9.5%. Then you can figure out, okay, well, we know that this is a bigger distance away or bigger recombination, so there's a higher chance that those are going to cross over and switch, right? So then we can start kind of mapping this out. So we can say one gene here between body color and wing length. That distance is further away because 17% were recombinant. But then if we add in, say, eye color and body color is 9%, so we can say that's maybe there. And then the antenna color and body color is 9.5%, so we can say that that gene is probably here. So from here to here is 9.5%, and this is 9%. And then we could do it again between wing length and eye color and start mapping all this stuff out. Okay, so that's how linkage maps work. The higher the recombination frequency, combination frequency, the farther the genes are on a chromosome from each other. From each other on a chromosome. Okie doke. That usually blows people's minds, so I'm just going to stop right there, and we'll pick up for the last bit in part 17, whatever we're on at this point.